live. Hello. Hi students, I think I am live. Hi everyone. Hi everyone, welcome to Unacademy NEET English. I'm your biology educator Ambika Sharma. I hope I'm clearly audible and visible to you all. Hi Bache Mishra, Naisha, Janu, Lokesh, Joshna, welcome to the session. Welcome everyone. So how are you doing I think after 2 to 3 days, I'm taking the session and I'm very much excited to start our new chapter, that is biotechnology principles and processes. And I hope you all have seen the video short, of course, right? Where I have explained everything to you. Here you people can see. Just watch this short after the class. So students, I have already taken the chapter molecular basis of inheritance. Right now I will be starting molecular biotechnology. So you know that all that 4 chapters are interrelated and they are very important for your NEET 2024. Isn't it? So excited to start the new chapter. Tell me quickly everyone, quickly. I want to see that energy in the chat section. Are you excited to start the new chapter? Quickly. And if you're new to our channel, you know what you have to do. You just need to click the subscribe button. So tell me everyone, I want to see that energy. And are you okay with the timing? Instead of morning, if I will start taking your session in the evening, is that okay? And trust me, these sessions will go together, right? There will be no break. Today we have part 1, tomorrow we have part 2, right? And I'm trying my best to finish it in 2 parts. Okay? Right? And then there will be the question practice. And finally, I will take 2 one-shots. After completing biotechnology principles and processes, biotechnology applications, I will be taking 1 shot of molecular basis of inheritance plus biotechnology all together. Okay, all together. And the questions will be practiced on our platform, on our Unacademy platform, where I will be taking the special class and that will be free of cost. Right? So you are going to get benefit. Right? Definitely you people are going to get benefit. So I want to see that energy in the chat section and quickly hit the like button, everyone. And see, that's my profile, right? Just download the Unacademy app or simply on Google type Unacademy. You can directly type my name, Ambika from Unacademy. Yeah, you will get this profile. Okay, subscribe to my profile and here you guys can see my free classes. Okay, you people can see my free classes. As of now, I'm taking Avengers batch, Avengers 3.0. If you want to join it, you can join it as well. And the coupon code will be Ambika10. But other than that, free classes are also there, bache. And see, I have scheduled one session. It is for 4th of November. Molecular basis of inheritance, and we'll be practicing last 10 years PYQs. Okay, so already you have the playlist for molecular basis of inheritance. Now the question practice, and once again there will be the 1 shot. Okay, once again there will be the 1 shot. So I want to see your energy there. So now let's start this chapter, that is biotechnology principles and processes as per your new NCERT. That's your chapter number 9, and as per your old NCERT, it's chapter number 11, right? Right. So let's start the chapter. Let's start this amazing chapter. And here you guys can see the free special classes only on the Unacademy app. And that will be from November 4th to 6th. So I already told you about my special class, and just subscribe to my profile. You will get all the details there, right? All the details there. And here you people can see, right? If you want to prepare for NEET UG 2024, we have the All India Test Series as well, for offline also, for online also, and for class 11 students also. I'll share the PDF in the Telegram group, and everything is written here. You can read it, you can use it, right, bache? So just scan these codes and you will get all the details. Clear, bache? All the details. The complete NEET 2024 preparation kit will be provided to you. So now let's start the chapter, that is your biotechnology. So if there is any doubt, do let me know, students. Yes, if there is any doubt, do let me know, related to any codes, related to anything. Quick. Sarat, that will be on 4th of November. Just subscribe to my profile. You will get all the details. So, biotechnology. In simple words, what is the meaning of biotechnology? It's simple. Biology means you are talking about the living organism. Technology, technique. So, here we are learning how can we use the technique, the technology to get best out of the living organisms. Honestly, here we are not doing something very good for other living organisms. Basically, the technique and the technology is used, right, so that we can get benefit from other microorganisms, and obviously, there will be the benefit of what? There will be the benefit of what? Human beings. Okay. So one student is asking here that, ma'am, what about the evolution? But evolution will be completed as well. I am not going to leave any chapter. I know I have to complete body fluid and circulation. I know I have to complete evolution as well. But in separate session, it will be completed. Right? So whatever is committed, that will be fulfilled. There is nothing to worry about. Fine, there is nothing to worry about. So there is, what is it? It is the biotechnology. So it deals with the techniques of using living organism or enzymes from organisms so that we can get what? We can get best out of it, isn't it? So that we can get best out of it. So ultimately it is for what? It is for the human welfare. So I'm not writing the definition here, the simple definition here. So I hope you all can understand this, that ultimately technology is used to get best out of the living organisms. Clear, bache? Now, when it comes to the biotechnology, there are two things. One is the old biotechnology, the traditional one. Right? Listen to me. I will make each and everything easy for you. Okay, that is a traditional approach. What type of approach it is? Yes, bache, I want to see the energy in the chat section, everyone. Right? And another is the new, or you can say that modern biotechnology. There are two things. One is the old biotechnology and another one is the new or the modern biotechnology. Now, what is the meaning of this, ma'am? See, when we use the word old biotechnology, here you are talking about the natural capability of the organisms. Right, Sarath? What are we discussing here? Here, we are considering the natural abilities of living organisms. I'll quote one example here. Natural abilities of what? Natural abilities of living organisms. Right, bache? For an example, if you talk about the penicillin, all of you know about the penicillin? That's a fungus, right? No, that's an antibiotic. What is penicillin? That's a antibiotic, isn't it? So we used to get this antibiotic from, right? We used to get this antibiotic from a fungus, and the name of that fungus is penicillium, right? Penicillium species, or you can simply say Penicillium notatum. So that's the natural capability of this fungus, that it can do what? It can form the antibiotic. Clear, bache? So that's the natural ability. Okay, so that comes under what? That comes under the old biotechnology. And I hope you remember, it is penicillin is the first antibiotic to be discovered. Clear? And moreover, let's talk about the yeast, which is a unicellular fungus. And from yeast, ethanol can be obtained, isn't it? So all that things, right? All that things, they come under the natural abilities, like lactic acid, right? Lactic acid bacteria that can form the curd. Okay, so that comes under what? Old biotechnology, the traditional ways. Now, when you talk about the new or the modern biotechnology, it is based on, bache, recombinant DNA technology. Here, what are we going to study? We'll be studying recombinant DNA technology. Basically, here we are going to talk about the genetic engineering, isn't it? What are we going to discuss here? What are we going to discuss here? We'll be talking about the genetic engineering here. That comes under the new or the modern biotechnology. Is that clear? Is that clear? For an example, here, if I have to quote one example, let's say the insulin, right? The gene from the insulin. The gene for the insulin can be synthesized externally by using the E. coli bacteria. Okay, human insulin gene can be synthesized outside the human body in the lab conditions by using the E. coli bacteria. So such examples come under the new or the modern biotechnology. Is that clear? So these are the two aspects of the biotechnology here. Done, bache? Done, bache? Is that clear? So if I talk about the definition, the proper definition, come to this point, right? We will be discussing NCERT as well. Okay, we are not going to leave NCERT, and this session is strictly based on NCERT. Okay, this chapter is strictly based on NCERT. Yes, bache, ready? I have covered this chapter. Okay, so now see, as per EFB, what is EFB? European Federation of Biotechnology. It is one MCQ. Okay, it is important. That's important. So EFB, what is EFB? It is European Federation of Biotechnology, and it has a given definition of biotechnology that encompasses both traditional view and modern molecular biotechnology. Here we are considering both the things. Here we are considering both the aspects. So if in simple words, I have to explain you the definition of biotechnology, so it's simple. We are integrating. What are we integrating? Yes, bache, we are integrating. What are we integrating? We are integrating the natural sciences, the technique, right? So that, right, we are integrating the natural sciences, the techniques with these living organisms, right? So that we can get the best products in simple way, if I have to say, right? In a very simple way, if I have to say, so basically it is the integration where you are integrating natural sciences, technology, right? So that you can get best products. Simple. In very simple words, let's read it from here. The integration of natural sciences and organisms, cells, parts thereof, and molecular analogs for products and services. Okay, for products and services. This is that technical language. Clear, bache? So ultimately, yes, Sarath is right. It should include what? Benefit. Benefit of what? Mankind, of course. Isn't it? Of course. So is that clear? Is that clear? So this is the definition. So this part is important. MCQ can come from this part as well. Okay. So now, bache, what is the next part? Next part is the principles of the biotechnology. Next, what we need to discuss? We need to discuss the principles of the biotechnology, and trust me, it is also very simple. So as I said, the session is strictly based on NCERT. We have that NCERT snips, right? With the help of that also, I will explain you the things. Ultimately, it's the NCERT that you need to master, students, and trust me, if you have command over the NCERT, you can easily score 300 plus marks in biology, right? If you follow proper strategy, like the sessions, right, the concept should be clear, proper revision should be there, MCQ practice you have to include, then trust me, it's easy for you people to get 360 out of 360 as well. You just need to follow a proper strategy. I know you must be thinking, ma'am, it's not easy to score 360 out of 360. I know, but if you have decided and if you are following that path, then definitely you can. And trust, and tell me, who's interested in scoring 360 out of 360 marks in biology? Quick, who's interested in scoring 360 out of 360 marks in biology? I want to see that in the chat section. Quick, everyone. So the next topic that we are going to discuss is principles of biotechnology, right? Principles of biotechnology. So here, as I said, two things are there. First is genetic engineering. Okay, first is genetic engineering. Clear, bache? So here it includes, first of all, write down Paul Berg. He is the father of genetic engineering. You should know about it. Okay, who's the father of genetic engineering? Paul Berg. Paul Berg is the father of genetic engineering. But what he did, just a minute. He basically, what he did, it is the genetic engineering. First of all, let me explain you the meaning of genetic engineering, then I will tell you what Paul Berg did. But genetic engineering, here it includes the manipulation. Manipulation of what? Manipulation of what? Manipulation of the nucleic acid, manipulation of the DNA, manipulation of the RNA. How that manipulation can be done? Let's say naturally there is a sequence. Okay, ultimately this is what we know, that when it comes to the DNA is a polynucleotide chain. What is DNA? DNA is a polynucleotide chain. So if DNA is a polynucleotide chain, you know that there are certain bases present, right? Your adenine, your guanine, cytosine, thymine. So they are present, right? In a sequence they are present here. So if I'll be adding any other gene here, any other base here, so obviously I'm doing what? The manipulation, right? The manipulation. Yes or no? Yes or no? So when it comes to the genetic engineering, here basically it includes the it includes to alter the chemistry of DNA and RNA. This is the language of NCERT, right? What is happening here in genetic engineering? It includes the techniques to alter the chemistry of the genetic material, right? It includes the techniques to alter the chemistry of genetic material. And when we talk about the genetic material, students, what do we have? We have the DNA as well as the RNA, right? What do we have? The DNA and RNA. Is that clear? Yes or no? Is that clear? So that's what genetic engineering is, right, bache? And after, it is not the only step here that we just need to alter the chemistry of genetic material. No, that altered DNA that needs to be inserted in the host organism as well, right? That we need to insert that into a proper host so that it can replicate there, right? So that it can replicate there. Are you getting my point? Are you getting my point? So when it comes to the principles of biotechnology, two things are there. One is genetic engineering and another is your bioprocess engineering or bioprocess technology or chemical engineering. That's the second principle here, right? That's the second principle here. So imagine you have altered a DNA. Now you have to shift it in a host cell where it can replicate. So it's not like that you can, you have just decided you will pick up the DNA and you will insert there in the host. No, you have to maintain certain conditions. You have to maintain proper conditions. Isn't it? What you have to do, students? You have to maintain certain conditions, or you can say that infection-free conditions, right? There should be a proper space where there is no contamination. Yes or no? Yes or no? But you will get the strategy video from November to May. What you should do, I will put a strategy video as well, right? Secondly, those who are saying that yes, we are interested in scoring 360 out of 360, the one and the only thing is complete your syllabus first. Your concept should be clear and everything should revolve around the NCERT. You need to master NCERT. Clear, bache? You need to master NCERT. Done, bache? Done, bache? So ultimately in genetic engineering, we are altering the chemistry of genetic material, right? That is the DNA and RNA. Then it will be inserted in the host where it will be changing the phenotype of the host, the physical appearance of the host, because you have modified the DNA. Okay, you have modified the DNA. Clear, bache? Clear, bache? It's simple. See, I will use the molecular basis of inheritance here, the basics of molecular basis of inheritance here. You have the DNA will form mRNA, and mRNA is going to form the protein, the enzymes. This is the central dogma. This is the flow of information. That is the central dogma. So DNA is forming mRNA, transcription. mRNA is forming protein, translation. And that's how a gene expresses itself. A gene is going to make the protein, that enzyme. That's how a gene expresses itself. That's what you need to keep in your mind. So if I'm going to alter this now, obviously I will get the altered product, I will get the altered protein, and accordingly that phenotype will change. That physical expression will change. Are you getting it? Are you getting it? Yes, bache. So next is, as I said, bioprocess technology or the chemical engineering. And here, what are we going to discuss? Here, it includes the maintenance of sterile ambience. What it includes, bache? It includes the maintenance of sterile ambience. Sterile means it should be free from contamination. Okay, it should be free from contamination. So you should have that environment where the environment which is infection free, right? Where you can further proceed. Okay, if I have to insert a modified gene into a host now, I need infection free condition. Clear, bache? So that comes under the bioprocess technology or the chemical engineering. Clear, bache? That comes under what? Bioprocess technology and the chemical engineering. So these two are the principles of biotech. Okay, and as I said, who's the father of genetic engineering? Paul Berg. So what he did, bache, he basically transferred the gene from a virus, right? The virus name was SV40, right? SV40 virus. From this virus, he took the gene. Okay, he took the gene, he put that gene in a bacteria. Of course, that is a world famous bacteria, your Escherichia coli, E. coli. Clear? What he did, he just transferred a gene from SV virus to E. coli with the help of lambda phage, right? With the help of a bacteriophage. Imagine, he picked up the gene from a virus, used another virus, and with the help of that virus, he shifted the gene to a bacteria. Okay, to a bacteria. That's why we say that Paul Berg is the father of genetic engineering. Understood? Paul Berg is the father of genetic engineering. And again, as I said, we are not going to leave the NCERT, so everything is written here in NCERT as well. So, principles of biotechnology includes genetic engineering and bioprocess engineering. So, it includes maintenance of sterile ambience in chemical engineering process to enable the growth of only the desired microorganisms in large quantity for the manufacture of products like antibiotic, vaccines, enzymes, and etc. So it's very simple. Guys, is there any issue with the voice? Yes, some students are saying, ma'am, increase the volume. Is there any issue? Yes, tell me. I think there is no issue, right? Subhashini, it's clear, right? It's better if you increase. I cannot, because from my side it's all good. Literally from my side it's all good. You please try to refresh once. Okay, so let's go. So see this. See, I'm going to alter the DNA. It's not a very easy process. It's not like that I have decided that today I'm going to cook something and I'll go to my kitchen and I have all the ingredients and I'll start cooking. No, it's not as simple. Okay, it is not as simple as we are reading, right? I'll tell you the detailed process. I'll tell you everything related to this, right? You will get to know then. So basically, students, what am I trying to say here is, see, altering DNA is not very easy. Imagine you have a cell. Okay, let's say it is a eukaryotic cell. Okay, now from the cell, you know that the DNA is present within the nucleus. So from the first of all, you have to figure out, right, from which cell you want to take the DNA. You have to isolate that cells first. Then from that cells, you have to isolate your DNA. And then from that DNA, you have to decide, you have to figure out that which gene you want, right? Which gene you want. Now we are telling it in a very simple way, but it is not that simple. Okay, it is not that simple. So it is going to take time. Okay, you need technique for it. So if we are doing such a process, so obviously we need certain results. Okay, if you are preparing for the NEET examination, if you are working day and night, why? Because you want to be the doctor, right? You want a seat in a good MBBS college, right? You want to serve people in that way, isn't it? You want to serve people in that way, isn't it? Isn't it, Kavin? I cannot increase the volume, right? I think there is some issue with your system. From my end, it's all fine, bache. Clear? So basically, if I am altering the DNA, it means I need that altered protein. I need that recombinant protein, that enzyme. Okay, that is what I want. And for that protein, for the synthesis of that protein, this chemical engineering, this bioprocess engineering, this bioprocess technology is very important, because it will provide me the infection-free conditions, right, bache? It will provide me the conditions, the technique where I can grow my product, right? Where I can grow my desired product. So that's what you need to understand. Is that clear? That's what you need to understand. So this is about the principles of biotechnology. Clear, bache? This is about the principles of biotechnology. Now, how this biotechnology came into origin, or you can say that what were the basis of that. Now you know the principle, genetic engineering and bioprocess engineering. But what made scientists think that we can do something like this? Isn't it? That we can do something like this. So two discoveries are there. What is there? Two discoveries are there, and you can see that these are the foundations, right? You can say that the foundation of biotechnology is dependent on two discoveries. Let me write it properly. Foundation of biotechnology depends upon two discoveries. Are you getting it? It depends upon two discoveries. One is obviously the restriction endonuclease and its use in vitro, right? It is not just restriction endonuclease. The best point is that we can use this enzyme outside, right? That restriction endonuclease that can be used in vitro. In vitro means outside the body in lab conditions. Secondly, right, this is the first point. Foundation of biotechnology, as I said, it depends upon what? It depends upon two discoveries. So the first is restriction endonuclease that can be used in vitro, and second is the plasmid, right? Plasmid which is present in bacteria. Let me explain this part first. Okay, let me explain this part first. So basically we are going to start with the Herbert Boyer, right? How are we going to start? We are going to start with the Herbert Boyer. Let me explain. So bache, in 1969, Herbert Boyer, he was studying the restriction endonucleases of E. coli. So I am going to explain you a story now. Okay, what am I going to do? I'm going to explain you a story now. So what are we talking? We are talking about the Herbert Boyer. Obviously, he's a scientist. So in 1969, what he did, he was studying the restriction endonucleases of E. coli. What was he doing? He was studying the restriction endonucleases of E. coli. Now, what he studied, bache? He studied that these enzymes, right? These enzymes, they can cut DNA at a specific site. What these enzymes can do? They can cut DNA at a specific site. That's the first point. Clear? That's the first point, Ashwat, that it can cut the DNA at a specific site. And moreover, when these restriction endonucleases, when they are cutting the DNA, you know, they are making some sticky ends. What is the meaning of sticky ends, ma'am? We don't know that. Please explain, right? I'll explain it. Just allow me some time. So what he studied, he was studying the restriction endonucleases, or you can say that restriction enzymes of the E. coli, right? He observed that these enzymes are having capability to cut DNA at a specific site, at a particular site, okay? And after cutting that DNA, these enzymes are generating certain sticky ends. You can say that these ends can clip with another DNA. Let me give you one example. Let's say, right, I'm giving you a very random example as of now. Let's say let's say this is a DNA that we have. It is having the bases. Okay, it is having the bases. It is having the nitrogenous bases. So obviously the sequence of ATCG will be there, right? The sequence of, yes, bache, the sequence of Sarath, Lakshmi, Kevin, Ashwat. Bache, I want response from your side as well. I want that energy as well. So basically the sequence will be there, and that sequence will be of ATCG, isn't it? The sequence will be of ATCG. So what is happening? That these bases are present over the DNA, right? These bases are present over the DNA. Now what's the point? That these enzymes are cutting DNA in such a way, right? These enzymes are cutting DNA in such a way that you are getting certain overhangs. Overhangs as in like, see this. It will be cut in this way that here you will be getting the overhangs, the clipped ends. Let's say here you are having certain bases. So you know that this ATCG can join with someone else also, right? This ATCG can join with another DNA, okay, which is complementary to it. Let's say here you have C, G, A and T. Then obviously it can join. So that's what he observed, okay? Okay, as of now, he's not aware that how can he use it, but this is what he studied. This part clear? Yes, bache, Joshna, this part clear? Sure? Sure. Right? Now we will talk about the another scientist, and his name is Stanley Cohen. What is the name of that scientist? The name of that scientist is Stanley Cohen. Bache, he was studying the plasmid. Okay, he was studying what? He was studying the plasmid. He found that plasmids, they are basically floating in the cytoplasm of, right, in the cytoplasm of bacteria. That's what he discovered. And these plasmids. They are kind of ringlets. But before explaining you the discovery of the Stanley-Cohen, let me remind you what exactly is the plasmid. Do you know what are the plasmids? Yes. Do you know what are the plasmids? Yes, students, quickly tell me. Quickly. Do you know what is the plasmid? I am going to explain it to you. But before that, whatever you know about the plasmid, quickly tell me in the chat section. I don't want students who just came here and just watch the lectures like this. If I'm asking something, you have to answer. Right? You have to answer. Very good. The session should be interactive. Exactly, Nandini, Vijay, Subhashini. Very good. So let me explain this. You know that when it comes to the bacteria, they are having their main DNA in the cytoplasm, and we used to call it as nucleoid, right? Because it is devoid of histone proteins and there is no nucleus in the case of bacteria. But in the case of bacteria, you see, right, that in the cytoplasm, you are having extra chromosomal DNA. What do you have there? You are having extra chromosomal DNA, and that is known as the plasmid. And this is, right, its replication, the replication of this plasmid is independent of, it is what? It is independent of the chromosomal DNA. It is independent of the chromosomal DNA. Are you getting it? So what am I saying? I'm talking about a bacteria. I'm telling you that in a bacteria, in its cytoplasm, you will see extra chromosomal DNA. Of course, it's a DNA. It is also going to have some genes, and that genes are going to give this bacteria special properties. First point clear? Everyone here in the class. First point clear? Second point is that this plasmid, its replication is independent of the chromosomal DNA. Okay, its replication is independent of the chromosomal DNA. That is, this plasmid is having its own origin of replication sites. We will discuss it in detail later. Okay, it is having its own origin of replication sites. Because of that, it can replicate by its own. So Stanley-Cohen, he was studying these plasmids, right? These ringlets of the DNA which are floating in the cytoplasm of bacteria, and he found that he discovered a method by which he can, you know, take out these plasmids, and then he can reintroduce that plasmids into another bacteria. This is what he did. Okay, Mishra, this is what he did. So basically what Cohen did, he developed the method, right? He developed method to remove the plasmids from a bacteria, to remove the plasmids from a bacteria, and then to reintroduce it into another bacteria. Got it? Into another bacteria. So these two discoveries were the foundation of the biotechnology. Clear, bache? These were the foundation of the biotechnology. Now, this is about the discoveries of Stanley and Cohen. Then, you know what they did? Do you know what they did then? Yes? Anyone here? Anyone here? So they combined the segments. Bache, what they did? They combined this process. Okay? Yes, bache? Do you know what they did? Yes. So basically, Stanley and Cohen, you know, they combined. What they did? They combined the DNA fragments. They used the DNA splicing as well. You know, the DNA splicing, removal of introns, but that will be done in the case of eukaryotic cell. In the case of prokaryotic cell, we do not have this process, the RNA processing, right? RNA processing is only there in the eukaryotic cell, which includes the removal of non-coding segments, introns from the DNA or mRNA, and then to add cap to do the capping and tailing of the mRNA. Okay, so basically your Cohen and Herbert Boyer, they combined their discoveries. They combined their discoveries, and what they did, they inserted the DNA segment, the desired DNA segment with the mother one. Clear, bache? Clear, bache? And after it, you know, obviously they were just conducting the experiments. So after it, you know what they did? Do you know what they did? They basically formed the first recombinant DNA. What they did? They formed the first recombinant DNA, and that's what we need to discuss. But before that, right, before that, we need to discuss something else. Okay, before that, we need to discuss something else. So this part clear? This part clear? So now come to this. You can read it from NCERT as well. We have to master NCERT also now. So we have completed one topic, and now we will move to the NCERT. Is that okay for all of you? Let's complete one topic and then let's relate that topic with the NCERT. Is that okay? If it is a yes, quickly let me know in the chat section. Yes, everyone. If it is a yes, quickly let me know in the chat section. Quickly, everyone. The energy should be there. The energy should be there. Strange, Dr. Strange, we will also discuss that. Let me start the sticky ends part, then you will get to know about it, okay? Okay, so see, you probably appreciate the advantage of sexual reproduction over asexual reproduction. When it comes to the sexual reproduction, you know that variations are there, right? Variations are there because of sexual reproduction, and you know why? Because there will be the crossing over. Do I need to explain it again? I think no, you know about it, right, bache? So the former means the sexual reproduction provides opportunities for variations and formulation of unique combinations of genetic setup, isn't it? Isn't it? And some of it which may be beneficial to the organism as well as to the population. But when it is the asexual reproduction, it is going to preserve the genetic information as such, right? There will be no alteration, there will be no variations because there is no gamete formation here. Clear, bache? So when it comes to the sexual reproduction, variations are there, but when it comes to the asexual reproduction, it will preserve the genetic information as such. Okay, so here they are explaining that when it comes to the traditional hybridization procedures, you know, not traditional hybridization procedures, right? I always give you the example of arranged marriage, that we are going to decide the superior plant varieties. So when there is a male plant, okay, when there is a bisexual flower, the male part will be removed, okay, and then the bagging of that flower will be done, and we are going to, when the stigma will be receptive, we are going to shed the pollen grains of the desired plant. Okay, okay, that comes under that traditional hybridization experiments in the case of flowers, in the case of plants. And when it comes to the animals, again the same way, two superior parents, right, superior male and female, they will be hybridized. Okay, their gametes will be fused. Clear, bache? So when it is the traditional hybridization way, right, it can often lead to inclusion and multiplication of the undesired gene. Basically here in this paragraph, what are they trying to explain? I already told you about the discoveries. Okay, I already told you about the foundation, isn't it? Isn't it? Here they are simply trying to explain that why can't we just rely on hybrids? Why do we need genetically modified organisms? That's how you can draw the comparison here. Okay, that what is the need to have the genetically modified organisms when we have the traditional hybridization ways? So according to you, why is it so? Why is it so? Yes, see, when it comes to the traditional ways of hybridization, when you are getting the hybrid, let's say this is a superior, these are the superior parents. Let's say this is a superior female or male. So when you are hybridizing, obviously that hybrid will have the characters of both, right? That hybrid will have the characters of both. So in the case of hybrid, bache, right, no doubt the desired characters of parents will be there, but undesired characters are also going to pass, isn't it? Here you cannot put any restriction that I want character A from plant to pass to the next generation and I want this plant to pass character B only. No, if desired characters will pass from one generation to another, undesired characters will also pass. Yes or no? Undesired characters will also pass. This is the first point when you are talking about the hybrids. Okay, secondly, when it is a genetically modified organism, what you have to do? You just need to alter the chemistry of the DNA, RNA. Then you can insert or even delete your desired DNA. Okay, it's It's as per your desire. Clear, bache? It's as per your desire. So you can insert and delete your desired DNA here. So it's not about the introduction of a new trait. You can also delete the trait that you don't want, right? So in the case of genetically modified organisms, when you use the genetic engineering, right? So there is no need to pass undesired traits. You can just remove that. Clear, bache? You can just remove that. That's why this modern biotech is better than the, it is better than the traditional hybridization, right? Right? So basically here they are saying that when it comes to the traditional hybridization procedures, right, they can often lead to inclusion and multiplication of the undesired genes along with desired ones. So basically with the desired characters, undesired characters will also be passed to the next generation. That's the meaning here. Clear, bache? That's the meaning here. And when it is the recombinant DNA technology, which is also going to use gene cloning and gene transfer. So mention it in a sequence. Your recombinant DNA technology, it is going to use your gene cloning. Desired gene will be cloned and gene will be transferred. But Sarat, I explained you the meaning here. Okay. So this technique is used to overcome the limitation of this hybridization process, and the undesired genes will not be passed to the next generation. Clear, bache? So now look at this part and then I will explain you the complete procedure here. Here, you know, the things are little, basically, I am following the NCERT only, but in a proper sequence, I'm going to teach you this chapter. Okay, bache? So here you can see the basic steps in genetically modified organisms, or if you want to modify an organism, what are the steps you need to take care? Okay, so now listen to me. In the NEET 2023 examination also, question was there from these parts, okay, from the steps of the genetic engineering. So when you read the NCERT, please mark everything properly, okay? And when you read NCERT, do not be like you're watching TV, you're watching movie, or you are scrolling the Instagram and then you are reading NCERT. No, while reading NCERT, be attentive, okay? Be attentive. Do not miss anything. Do not miss anything. You should be focused on your book only, okay? Stop multitasking at that time. Done? Stop multitasking at that time. So here you guys can see the three main steps in modifying organisms genetically. So the first is you have to identify the DNA with desirable genes. Right? What you have to do? You have to identify the DNA with the desirable gene. Like this is a particular organism. This organism is having this particular character and I want that. Okay, I want that. I want to propagate that gene. I want to get that particular protein. So firstly, you need to figure out that. Then after figuring out that, you have to introduce that identified DNA into the host. What you have to do? You have to introduce the identified DNA into the host. And after that, bache, there should be the maintenance of introduced DNA in host and the transfer of DNA to its progeny. So these are the three steps. Clear, bache? These are the three steps. Done, bache? So firstly, identification of DNA with desired gene, then introduction of that identified DNA into the host. So you should have a competent host as well. We will talk about it as well. So then maintenance of introduced DNA in host and transfer of DNA to its progeny. So obviously here you need the chemical processing or the bioprocess technology. Understood? Done? Okay. Now let's talk about the tools, and the tools will explain you the entire process. Done, bache? So please make the notes as well. Fine. Please make the notes as well. So what are we going to start? We are going to start the tools of the recombinant DNA technology. Okay, tools of recombinant DNA technology, and it includes, the very first thing is enzymes. Okay, the first thing that we need to read here is enzymes. So what do you know about that enzymes? Yes, the very first thing that we need to know is enzymes. Yes, tell me, we are talking about the tools of recombinant DNA technology. First is enzymes, then yes, gel electrophoresis. That's what we need to study, right? These are the tools of recombinant DNA technology. So gel electrophoresis is there. Then comes bache vectors. Then DNA delivery system. That how can we deliver the desired DNA to the host. So first is enzymes, then is the gel electrophoresis, then comes the vectors, then DNA delivery system, right bache? And then finally what do you need? You need the host, right? And for that we will use the word competent host, the host which is capable of taking that desired DNA. So these are the tools for the recombinant DNA technology. Enzymes, right bache? After enzymes, what should be there? Gel electrophoresis. We will discuss that in detail, right bache? And the third thing that we need is the vectors. These are going to behave like the gene taxis, which will be used to transfer the desired gene from one host to one organism to the another. Then DNA delivery system like electroporation, right? We will discuss that also in detail. Fine. And then finally the competent host. So that's what we have to cover. So the first thing is enzymes, and here there are various type of enzymes that we need for the biotechnology, right? There will be the lysing enzymes, there will be the cleaving enzymes, there will be the enzymes that will behave like the glues, right? There will be the enzymes which will be used for the synthesis. So we need various type of enzymes, bache, right? We are going to need, we will be needing different types of, yes bache, different types of enzymes. And here I will tell you about the restriction endonucleases as well. Clear bache? Clear bache? So that's what we are going to cover here. Okay, so the very first thing that we need to know here is the lysing enzymes. Which enzymes? The lysing enzymes. Bache, what is the meaning of this word? To lyse something, right? Like we know about the hydrolysis, right? Photolysis. Lysis is the word, right? Lysis is the word. Lysis means, yes bacho, it is related to, quickly tell me in the chat section. To break, exactly, right? It means breakdown, to break something. Of course, you are right. Okay, it means breakdown, to break something. Now bache, if you are talking about the DNA, that you need DNA from the host, or you need DNA, you need to figure out the desired DNA, right? Right. So firstly, you should know now that which DNA you want, which gene you want, and after knowing that, you have to extract that, you have to get that, isn't it? Isn't it? So basically, let's say this is a cell, this is a eukaryotic cell. Let's say it's a fungal cell. What is it? It is a fungal cell. Now from this fungal cell, you have to take out your desired gene, desired DNA. So firstly, you have to break the cell wall here. Yes or no? Firstly, you have to break the cell wall here, right? Then only you'll be able to reach to that nucleus and then to extract the DNA. So that's why the very first category here is the lysing enzymes. Okay, the very first category here is the lysing enzymes, and these are the enzymes which are used to open the cells to get the DNA. Clear bache? These are the enzymes. These are the enzymes that are used to open the cell to get DNA, isn't it? To get what? To get DNA, isn't it? Yes or no? So let me give you one example. We started from the fungal cell. So can you tell me what is there in the fungal cell wall? What is there in the fungal cell wall? Fungal cell wall is made up of exactly. Fungal cell wall is made up of what? Fungal cell wall is made up of chitin. It is the fungal cellulose, if you remember. Okay, it is the fungal cellulose, if you remember, chitin, right? It's a homopolymer. Homopolymer of, yes, revise it quickly. It's a homopolymer. Homopolymer of N-acetyl glucosamine. Okay, it's a homopolymer of N-acetyl glucosamine. So if I have to take the desired gene from the fungal cell, means I have to open the fungal cell. So I have to break this, right? I have to break the cell wall. So which enzyme I'm going to use? I will use chitinase. Okay, that will act on this chitin. So which enzyme I'm going to use? I will use chitinase. Okay, I will use chitinase. Clear, bache? Right, right. So fungal cell is digested by, fungal cell wall is digested by chitinase. Okay, it will be digested by chitinase. Done. Now the second point is, let's say if it is a bacterial cell, then which enzyme you can use? Lysozyme. Okay, which enzyme you can use? Lysozyme. So it is used to digest cell wall of bacteria. Okay, it is used to digest cell wall of bacteria. Clear, bache? And then cellulase. What is the name of the enzyme? It's cellulase. So obviously it is used to digest cellulose of plant cell wall. Clear, bache? It is used to digest cellulose of plant cell wall. So that's why these enzymes we need to use first, and these are the lysing enzymes. Is that clear? Yes, bacho. Is that clear? Right? Please make the notes as well. Please make the notes as well. Okay. So next category of enzyme is cleaving enzyme. Right? They are going to cleave the genetic material. Now see, imagine the process as well. You have a cell, you are going to open that cell, you are going to break its cell wall. Okay, you need the lysing enzyme. So after breaking that cell wall, you need your DNA. You need to break that DNA, right? You need to get the desired sequence. So that's why you need the cleaving enzymes, right? So you are, yes. Now you can also relate this process. You can also understand it in an easy way, right? So next category is of what? Next category is of cleaving enzymes. Next category is of cleaving enzymes. So they are used to cleave the DNA. Clear, bache? They are used. Okay. So they are used to cleave the DNA. And here you have two categories. Okay, the one is exonuclease. So basically you are talking about the nucleases here. The another enzyme is endonuclease. But DNA is a nucleic acid, and when you are using the word nuclease, means it is going to digest nucleic acid. Okay, it is going to digest nucleic acid. So when the word is exo means outside, or you can say the terminal ends, right? Let's say if this is a DNA, it's a linear DNA, right? So in this DNA, these are the terminal ends. These are the terminal ends. So these ends are cleaved with the help of which enzyme? With the help of exonucleases. Which enzymes are going to cleave these ends? Exonuclease. Exo means outside. So that these nucleic acid digesting enzymes, they are going to break the ends. Clear, bache? They are going to cut the ends. And now when the word is endo means inside. So these enzymes are going to cut the nucleic acid at any part other than terminal ends. Okay, they can cut the DNA at any part other than terminal ends. Means these endonuclease can cut the DNA here, here, or here. So anywhere leaving the ends. This part clear? Yes, bache. Exonuclease, endonuclease, this part clear? Yes. Is that clear? So exo and endo. So if I have to differentiate, see, one point is this: terminal ends and anywhere other than terminal ends, right, bache? Anywhere other than terminal ends. Now when you talk about the exonuclease, you know what that exonuclease is going to do? Yes, bache. This exonuclease, it can cut the, wait, I'll write it here, just a second. Endonuclease and here exonuclease. So see, when you are talking about the exonuclease, it can cut the single strand of DNA, single strand of DNA, or the gaps, or it can cut at the gaps in between the DNA, right? When it is the exonuclease, it can cut on single strand of DNA or gaps, right? Or if there are gaps in double strand of DNA. Let's say that's the DNA sequence that we have. So if there is a, so basically exonuclease can cut here. Okay, exonuclease can cut here the single strand of DNA. Exonuclease can do what? Can cut here the single strand of DNA. And moreover, if let's say it's a double stranded DNA and there is a kind of gap in between, okay, let's say here in this portion the bases are missing or there is a gap, then it can make the cut here. Okay, then it can make the cut here. So basically when it comes to the exonucleases, right, so exonucleases are not cutting the DNA in this way that we can use it for genetic engineering. Okay, so you are not using the segments that are cut by exonuclease in the genetic engineering. Is that clear? Sure, bache. So when it comes to the exonuclease, the separated fragments cannot be used in the genetic engineering. Okay, the separated fragments cannot be used in genetic engineering. Understood? Now, you know about the endonuclease. It can cut anywhere other than terminal ends. So this endonuclease can even cut the single strand or it can even cut the double strand of the DNA. Okay, when it comes to the endonuclease, it can cleave one strand or both the strands of DNA. It can cleave one strand or both the strands of DNA. So obviously it is used in genetic engineering. Done, bache? It is used in genetic engineering. And which endonuclease you are going to use in genetic engineering? Restriction endonucleases, right? Which endonucleases you are going to use in genetic engineering? Restriction endonucleases. Do you know why? Because these endonucleases, they cut DNA at specific sites. They are not cutting the DNA randomly. They are not just cutting the DNA at the ends only. So they are cutting the DNA at a specific site. That's why we are using it in the genetic engineering. So the next topic is your restriction endonuclease. Okay, so firstly endonuclease. So here you will be talking about restriction endonuclease. These are the special type of endonucleases which are going to cut the DNA at specific site, and we used to call them as chemical scalpels or the molecular scissors. They are also known as molecular scissors or chemical scalpels. Done? Clear? So endonuclease, yes, bache, endonuclease, so it is a type of endonuclease, your restriction enzyme, or you can say that your restriction endonuclease. Done, bache? Okay. Okay, restriction endonuclease. So this is the topic that we need to discuss in detail. So question can come from this part. So kindly listen to me carefully. Okay, kindly listen to me carefully. So these restriction enzymes, they belong to a large class of enzymes called endonuclease. Already I told you. This line is given in NCERT. These restriction enzymes, they belong to a large class of enzymes, that is your endonuclease. Clear, bache? So what is their function? What is their function? Bache, see, these restriction enzymes, first of all, bache, they can be used in vitro. Okay, they can be used in vitro, means they can be used in the lab conditions. So they are going to inspect the length of the DNA first. They will inspect the length of the DNA. They will check that length of the DNA. Then they will recognize the specific sequence and then they will make the cut. Okay, what is going to happen in the case of these restriction enzymes? They are going to inspect the length of the DNA. They will figure out where is that special sequence that they can cut. They will cut that special sequence. Okay, then they will cut that special sequence. So that's what you need to know about these restriction enzymes, that they are used in rDNA because they can be used in vitro. Yes, these enzymes, they are used in rDNA, means used in recombinant DNA technology. In vitro, to recognize and cleave, to recognize and cleave the specific, right, specific sequence of DNA. Is that clear? The specific sequence of DNA. Is that clear? So it is used in vitro to recognize and cleave the specific sequence of DNA. Understood? And as per NCERT, this word specific sequence is very important. Okay, it cannot cut randomly. It will recognize a specific sequence and then only it will make the cut. Fine, bache, then only it will make the cut. So firstly, it will inspect the length of DNA sequence. Let me write it. Inspect the length of DNA sequence. Recognize a specific sequence. Then cleave the specific sequence to generate sticky and the blunt ends. Okay, it can cleave that specific sequence and then it will be forming the sticky ends or the blunt ends. Mainly it is the sticky ends. Clear, bache? Mainly it is the sticky end. Right? Right? And do you know that this is another important question? See why am I highlighting it? Because again, it was also there in the NEET examination that restriction enzymes can inspect the length of DNA sequence or not. So generally, when you don't understand the meaning, right, you are like, okay, restriction enzyme is something which will make the cut. Okay, it is not just going to make the cut. Firstly, it will inspect length of DNA. It will figure out where is that sequence that they can cut. Then they will cut. Okay, then they will cut. And do you know how that restriction enzyme will make the cut? Yes. Do you know how is it going to make the cut? This is another important question. See, it's very simple. When you talk about the DNA, you know that, right, that bases, nitrogenous bases are there in center, right? And then there is a sugar phosphate backbone. I hope you know about it, right? Then there is what? There is a sugar phosphate backbone, right? Yes, bache, do you know about it? There is a sugar phosphate backbone. Yes or no? Of course. Right, of course there is. So basically they cleave the specific sequence of DNA at their sugar phosphate backbone. Okay. So it is going to cut, it is going to make cut here in the sugar phosphate backbone. Are you getting it? Yes, everyone. Are you getting it? Yes. So it is going to break here in the sugar phosphate backbone. So it is another important MCQ. Don't miss this part. Fine. Don't miss this part. And now the another point is that which sequences are cleaved by the restriction endonucleases. I am using the word now specific sequence. Again and again, this is the word that I'm using. Specific sequence, right? So when you talk about these specific sequences, bache, actually they are the palindromic sequences. What type of sequences they are? They are the palindromic sequences having 2 to 8 nucleotides. For an example, what is the meaning of a palindrome? See, like you have a word madam, M-A-D-A-M. So if you are reading it from this side, M-A-D-A-M, even if you are reading it from this side, M-A-D-A-M. So spellings are going to be same. Yes, spellings are going to be same. Another word is dad, D-A-D, radar, R-A-D-A-R. So even if you are reading them from this side, you will find the same spellings and here also. right? Here also. So such sequences are, yeah, Malayalam, right? Such sequences are palindromic sequences. What type of sequences are they? What type of sequences are they? They are the palindromic sequences. Clear, bache? So restriction enzymes, they are, as I said, they will also recognize specific sequence. That sequences are palindromic sequences, and then it will break the sugar phosphate backbone. Then it will break the sugar phosphate backbone. Are you getting it? Then what will it do? They will break the sugar phosphate backbone. Understood? Yes, bache. And do you know how are we going to read that? I'll give you one example. GATTC, GAATTC. This is also a very famous palindromic sequence. This is the sequence recognized by the restriction enzyme EcoR1. We will discuss about this EcoR1, right? We are going to discuss about this EcoR1. So this is also a restriction enzyme. What is it? It is also a restriction enzyme. So EcoR1 recognize this particular sequence. It is GAATTC. You know, I call it as GATTC, right? It is my way to remember it. So you can simply call it as GAATTC. Okay. So in 5' to 3' direction, in 5' to, right, bache, in 5' to 3' direction, what is it? What is it? It's GAATTC. And here also, it's GAATTC. Clear, bache? So basically, when you are reading it from 5' to 3', so see GAATTC. Here also it's GAATTC. So such type of sequences are palindromic sequences. Clear, bache? Palindromic sequences. Right? Right? Palindromic sequences. Done? Understood? And always remember, right, we read these sequences from 5' to the 3' direction. How are we going to read it, bache? We will read these sequences from 5' to 3' direction always. You are never going to read it like this. C, T, T, A, G. No, 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 never. You will always read it in, how are you going to read it? In 5' to 3' direction. Understood? So basically, from where are you going to get the restriction enzymes? Restriction enzymes are obtained from bacteria. You are not going to get them from the eukaryotic cell. They are obtained from, yes, bache, they are obtained from bacteria. They are not obtained from any other eukaryotic cell. This is the first point that you need to remember. So, you know, why these restriction enzymes are present in bacteria? Can you tell me? What is the function of these restriction enzymes in the bacteria? Yes. Tell me quickly. Tell me. Actually, bache, in the year 1963, two enzymes were obtained from bacteria. Two enzymes were obtained from bacteria. Wait, I'll tell you. One enzyme was cutting. One enzyme was cutting the DNA, cutting the viral DNA. Okay, and another enzyme was adding the methyl group. That enzyme was doing the methylation. Right, now I'm telling you the story of these restriction enzymes. Fine, bache. I'm telling you the story of these restriction enzymes. So basically, as I said, that restriction enzymes, they are obtained from the bacteria, right? From where are they obtained? They are obtained from the bacteria. Clear, bache? So in the year 1963, two enzymes were isolated from bacteria. One enzyme was cutting the viral DNA. Now you must be thinking, ma'am, you are talking about bacteria. How come the viral DNA? I'll come to that. And another enzyme was adding the methyl group. So basically that enzyme was doing the methylation. Actually in bacteria, and it was studied in the case of E. coli, of course. This is our world famous bacteria. Hai na? We always talk about E. coli, Escherichia coli. Clear, bache? So actually in bacteria, we have one thing called restriction modification system or restriction modification enzyme. Whatever you want to say. in the bacteria. That's what we have. There is no need to write down this part, but you should understand it. Okay, there is no need to write down this part, but yes, you should understand this. So when we talk about the restriction modification system, here you are having two enzymes, right? Here you are having two enzymes. Of course, basically this system is having two components. One is the restriction enzyme and another is the methylating enzyme. One is the restriction enzyme and another is the methylating enzyme. So now you can simply understand, right, bache? Or methylating, or you can say that it is the modification enzyme. It is the modification enzyme. So you know that restriction enzyme which will make cut, modification enzyme which will add methyl group. Now I will tell you the function, right? What will I do? Now I will tell you the function here. So it's simple. See, let's say this is a bacteria. It is having the restriction modification system. Now there is a bacteriophage. What is it? It is a bacteriophage. But I'm just trying to explain you this part here. So please listen to me carefully. I just said that restriction enzymes are obtained from the bacteria. So restriction enzymes are the one which are going to recognize a specific sequence. So the question arises here that why that enzymes are not cutting the DNA of that particular bacteria. Okay, fine. This is understandable that we are getting the restriction enzymes from a bacteria. But my question is why that enzymes are not digesting the DNA of that bacteria. First point. Okay, so scientists, when they were studying all that things, they found the restriction modification system having two components. One is the restriction enzyme, another is the modification enzyme. So now what is happening, I'm telling you a story. In the form of this story, you will understand it. Okay, you will understand it properly. So there is a bacteriophage. Bacteriophage is what? It's a virus that infects bacteria. Yes or no? It is a virus that infects bacteria, and you know that bacteriophage is a DNA virus. It's a double-stranded DNA virus. Now, here in E. coli, let's say this is a E. coli, it is having its nucleoid. Okay, and this bacteriophage is trying to insert its own DNA here. Right, it is trying to insert its own DNA here. You know, na, that's how the viruses work. They do not have their proper machinery. Outside the body, they are just the crystallizable protein. They are just the inert protein. They need a living host. They need the machinery of that living host. Then only they can work. Right, then only they can work. They can divide. So bacteriophage is trying to insert its DNA into the bacteria and trying to use the machinery here. But here in the bacteria, we have one enzyme, restriction enzyme, that will not allow. That's how I'm going to explain it. So this restriction enzyme present in bacteria will not allow the viral DNA to grow here. So it is protecting the bacteria from the virus, not allow the viral DNA to grow in bacteria. Right? Why? Because this restriction enzyme will cleave its DNA, will cleave the bacterial DNA. This part clear? First point. Now, what is the role of modification enzyme then? Right? What is the role of modification enzyme then? It is also very simple. Fine. It is also very simple. See, I told you that these restriction enzymes are not affecting E. coli's DNA. Why? Because in this E. coli, obviously there is a sequence which can be recognized by this restriction enzyme. Right? There is a sequence in this E. coli's DNA also that can be recognized by restriction enzyme. But because of modification enzyme will add methyl group. It is going to add methyl group to that sequence of bacteria's DNA. Right? It is going to add methyl group to that sequence of, right, that sequence of bacteria's DNA that can be recognized by restriction enzyme. Got it? So if this sequence is methylated, then restriction enzymes cannot cleave it. Clear, bache? If that sequence is methylated, right, then the restriction enzymes cannot cleave it. Got it? Yes, tell me, is it clear? Is it clear? Okay, so bacteria itself is having restriction modification system. So restriction enzyme will not allow any other DNA to grow here in bacteria, and modification enzyme, they are protecting, right? They are protecting the equalized DNA from that particular restriction enzyme by adding the methyl group. Okay, by adding the methyl group. So if there is any doubt related to this topic, tell me quickly. Okay, tell me quickly if there is any doubt related to this topic. Done? Sure. So you know that first restriction enzyme isolated is your HIND2, isn't it? It is your HIND2. Come to the NCERT part. Let's revise it from NCERT. We are talking about the enzymes, right? We are talking about the enzymes. I told you about the cleaving enzymes. I told you about the restriction endonuclease as well. So here you can see, but restriction endonuclease is having types also. We will discuss that. But firstly, focus here. Okay, firstly, focus here. So see, the first restriction endonuclease is HIND2. It is HIND2. Another important MCQ. Whose functioning dependent on a specific DNA nucleotide sequence. You know that these enzymes, they can recognize a particular sequence. That's why here we are saying, okay, that's why here we are saying that the functioning of this enzyme is dependent on a specific DNA nucleotide sequence, and it was isolated and characterized 5 years later, right? It is isolated and characterized 5 years later. Okay, so HIND2 was discovered, but the sequence, right? The functioning whose functioning depended on a specific DNA nucleotide sequence, and it was isolated and characterized 5 years later. Okay, 5 years later. So this information is important. MCQ can come from this part. So it was found out that HIND2 always cut DNA molecule at a particular point by recognizing a specific sequence of 6 base pair. So yes, another important MCQ. Right, HIND2 is going to recognize how many base pairs? 6 base pairs. How many base pairs? 6 base pairs. Done, bache? So this specific base sequence is known as recognition sequence, right, bache? And you know that it is going to be palindromic. Okay, so the sequence recognized by your restriction enzyme, it is also known as recognition sequence. It is also known as recognition sequence. Clear, bache? Clear, bache? Okay, so in addition to HIND2, today we know more than 900 restriction enzymes that have been isolated from over 230 strains of bacteria. This part clear? Yes, this part clear? So here we have the nomenclature of these enzymes. Let's discuss the nomenclature and the various types and the detailed process we will discuss in next class. But firstly, let's discuss the nomenclature. And before nomenclature, remember your restriction enzymes, they are of 3 types. You have restriction endonuclease 1, you have restriction endonuclease 2, and you have restriction endonuclease 3. Do you know about it? Do you know about it? You have restriction endonuclease 1, 2, and 3. How many restriction endonuclease do you have? Restriction endonuclease 1, restriction endonuclease 2, and restriction endonuclease 3. But here, when it comes to the genetic engineering, restriction endonuclease 2 is used in genetic engineering. Why, bache? Because it can recognize the specific sequence. It can recognize the specific sequence of DNA and can cleave that, right? It can cleave that at its sugar phosphate backbone. But when it is the restriction endonuclease 2, it can only recognize the sequence. It cannot cut that sequence. That's why it is not used in genetic engineering. Even the restriction endonuclease 3 also, it can just recognize. It can inspect the length of DNA and can recognize. But restriction endonuclease 2, it can recognize as well, right? It can recognize as well, right? And it can even cleave the DNA sequence. Fine, that's why restriction endonuclease 2 is used. Fine, bache? Done? Okay, so now let's talk about the nomenclature. So see, I gave you the example of one restriction endonucleases, that is ECO-R1. So E here, what this E here is signifying? Yes, bache, what this E is signifying here? Tell me. Actually, the name of the enzyme is also from the prokaryote from where the enzyme is taken. Like if I'm taking this enzyme is taken from the Escherichia coli. That's why its name is also on the basis of that bacteria. Clear? So when you talk about the first letter here, okay, the first letter of the genus becomes the first letter of the name of the enzyme. Right? Here, first letter of genus become the first letter of enzyme. So that's why E here is Escherichia. Done, bache. It's the first name of the genus and it is the first name of the enzyme, right? And then first two letters of the species will make second and third letter. Now these second and third letter, what are they? They are the first two letters of species name. You know na, species name is coli, C-O-L-I. So CO, they are making second and third letter here, right? Second and third letter here. Clear, bache? Clear, clear, clear. So they are written in small letters. This is also important. It is written in capital letters. You have to write it like this. And it is written in small letters. Fine. It is written in small letters. Understood? Understood. So the next part is, right, bache? The next part is the fourth letter and it is the first letter of the strain. This fourth letter is first letter of strain, right? It is the first letter of strain. From, you know, na, E. coli bacteria is having different strains, right? That is RY1. So this R is telling us about it. Clear, bache? This R is telling us about it. It is the first letter of strain and it will be written in capitals. Okay, it will be written in capitals. And one more thing here, the eco, right? This part, these first three letters, we have to write it in italics. Okay, they will be written in italics. Clear, bache? They will be written in italics. Done, bache? And then you have the Roman number written at the end of the name. It indicates the order in which enzyme was, right? It indicates the order in which enzyme was isolated from that strain of bacteria. Okay, from that strain of bacteria. So basically it is in Roman letters. Fine. So this is about the nomenclature. You need to remember it. One question can come from this part as well. And sometimes, you know, after this enzyme, you are also going to see the, you know, the lines. It can be like this. Okay. Sometimes you will see it over the sequence. Sometimes like this, sometimes like this. So they will tell us that is it going to form the sticky end or the blunt end. So about the sticky end and the blunt ends, we will discuss in the next class. Clear, bache? So this eco R1 part is important. See, it is given in NCERT here. Okay, that eco R1 comes from Escherichia coli RY13. So we have to take the first letter of that strain, which is making the fourth letter, and then that one is showing the Roman number. So the strain is RY13 here, right? So R is derived from the name of the strain, and the Roman number following the name indicates the order in which the enzyme is isolated. Done? Clear, bache? So this part is covered. So see, it is also given in NCERT that each restriction endonuclease functions by inspecting the length of the DNA sequence. Once it finds its specific recognition sequence, it will bind to the DNA and cut each of two strands of double helix at specific points in their sugar phosphate backbone. So remember, it is the sugar phosphate backbone. Clear? It is the sugar phosphate backbone and the sequence is palindromic sequence. So here you have certain examples also. So rest, bache, we will discuss in the next class. Okay, we will discuss in the next class. So we'll try to finish it tomorrow. Otherwise, in 3 parts we are going to finish it. So this chapter is important. You know that, right? And the special classes on molecular basis of inheritance I'm going to take on the Unacademy platform, right? That special classes, they'll be free of cost. So in this way, you can revise both the chapters simultaneously. And you know that genetics and biotechnology all together, they are very important. Okay, all together they are very important. And bache, if you want to be the part of Avengers 3.0 batch, the link is given in description box. What you need to do, you just need to use the coupon code Ambika10. That's all. And you will be the part of my batch where you will get the amazing content from the amazing educators. Done? Yeah, that MCQ practice for last 10 years, that session is on 4th of November. Okay, that session is on 4th of November. Here you can see my profile. Okay, just subscribe to my profile and you'll, in free classes, you'll find this session. Molecular basis of inheritance, last 10 years PYQs. Clear? Last 10 years PYQs. Okay, so take care. Good night. Bye bye. See you soon. So see you all tomorrow at 8 p.m. Okay, so take care. Do subscribe to my profile. I'm also going to take another sessions as well, like the question practice sessions on the plant physiology, genetics, and I'm going to start the biomolecules as well. So see you all there. Take care. Bye bye. Thank you so much. And if you like the class, do let me know in the comment section. I will wait for your comments.